Dear students, this is Dr. Pooja Thakkar, a biology expert. This is our third session in cell biology on cell wall and gram staining. In the previous session, we have already discussed regarding the outermost boundary, that is the plasma membrane. And in this session, we are discussing regarding the cell wall, which is the typical structure, or you can say the speciality of typically a plant cell as well as fungi and certain other individuals. Okay, so it is present as the outermost coating in the plant cell as well as in the fungus. In case of alga, cell wall is also present, and it is made up of cellulose, galactans and minerals like calcium carbonate, that is giving stiffness to the algal cell wall. Cellulose is, you can say, the basic component of cell wall, any kind of cell wall, and galactan here is serving as a sugar moiety. Now in case of plants, as you see, cell wall mainly consists of cellulose, hemicellulose, pectins and other kinds of proteins. The other kind of variant of cellulose, that is hemicellulose, and a particular material which is giving the stiff as well as little sticky boundary, that is pectin, is also present. Now fungus is having a typical structure called chitin in the cell wall. So the cell wall is chitinaceous here. The primary cell wall, if we talk about young plant cell, the preliminary formed wall is called primary cell wall, which is thin and elastic, and it is having the low cellulose content, that is less than 20%, which is capable of growth and which diminishes in mature cells. I mean, when the cell undergo maturation process, these primary cell wall will undergo degeneration process because the secondary cell wall start to form. This secondary cell wall is formed on the inner side of the primary cell wall that has less hydrated condition, that is less water condition, and having high cellulose content, that is 20 to 40%, as well as it is having the hemicellulose, which is 25%. This secondary wall is made up of three layers, that is S1, S2 and S3, that also contains lignin in certain amount, which will reduces the hydration power, that will reduce the water content of that wall, and that will lead to increase in hardness of the wall. In this particular diagram, you will be able to notice the primary cell wall. In that, you will be able to notice green colored structure, pink color structure, that is hemicellulose, pectin, and the thread-like structures called cellulose microfibril, which are black in color. But likewise, when the primary cell wall will get shorten up, or you can say that becomes slightly reduced in size, and later it will get degenerated. At that time, secondary cell walls start to form. It is having lignin, which you can be able to see in the next diagram, that is B diagram. That is the rounded bulgy structures made up of chain. So those structures are called lignin. They are also having pectin at certain locations. They are having varieties of proteins, cellulose microfibril in typical pattern, as well as hemicellulose in the specific pattern with the cellulose microfibril. So it is comparatively very thicker and very stronger. Now, if we talk about bacteria, how we will identify as these kinds of organisms are gram positive or gram negative. So based on the type of cell wall nature that is present in the particular bacteria, according to that you can distinguish it in two cases, that is gram positive individual and gram negative individual. Now first, what you need to do is you have to take the slide. You have to take the bacterial culture from any of these sources. For example, if I'll give the example, you can say lactic acid bacteria, that is commonly found in curd. So you can have a spoonful of culture or a loopful of culture of that particular curd into the water. You have to make it diluted, and one of the specific diluted sample is taken to have the sample on the slide. Okay, so loopful of sample is taken on the slide, that is used to make a smear on it. Then the slide is typically put on slightly on the burner or to have the flame, so that that particular slide become heat stained, or you can say heat sterilized. Okay, now After that heat treatment, the bacteria will get fixed on the slide. Now you can take varieties of staining actions on that. So primary stain here we are using, that is crystal violet, or it is also called as gentian violet. You have to use a particular you can say a dropper to take the sample of that primary stain, and you have to put it on the slide. After a minute, during a minute, it will get penetrated into the cell membrane, which is typically made up of a peptidoglycan layer, as we have discussed in the first session. What you need to do then, you have to drain the stain, little wash the slide, and then flood the smear with the iodine solution. This iodine is here working as a mordant that will form the complex with the crystal violet stain or gentian violet to make the complex called CVI complex, so that they can fix the crystal violet at particular location in the cell wall. So overall, peptidoglycan in both the cases, either it is gram positive or gram negative, it will get stained. Now when you are applying decolorizer on that decolorizer is typically washing off each and every crystal violet particle from the gram negative individual, but they are not at all affecting any gram positive individual, because gram positive, as we all know, they are having multiple layers of peptidoglycan, and crystal violet iodine complex already has fixed into that particular area, so it will not get detached. So the gram positive cells will still appear purple, but the gram negative cell will now considered as the colorless cells. Now you can apply the counter stain, which is also called as secondary stain. These kind of counter stain is typically here we are using, that is safranin, which is staining the cell as red or pink. So gram positive bacteria are already purple in color, so they will not at all absorbing any secondary stain now onwards. But the gram negative bacteria, which are colorless right now, they are accepting the stain, accepting the secondary stain, and that is giving pink color. Okay, so here the actual process has been shown in the diagram. That is, step 1 is heat fixation, then step 2 is crystal violet, which is primary staining, then gram iodine, that is CVI complex formation, then the decolorization agent or decolorizer will work, that is alcohol, which is 95%, and then the counter stain. And then you will be able to notice the result, that is gram positive as blue or purple in color. You can say either they are cocci or bacilli, they'll show this kind of respective shape as well as color, and gram negative are basically pink or red in color. Okay, so overall result is gram positive are blue, purple, gram negative are pink or magenta or red. So here I have just for the knowledge side given you the bacterial infection condition, that is gram positive and negative both are responsible for causing severe diseases. Gram positive cocci infection, best example is staphylococcus aureus, which can cause skin infections in varieties of cases. Likewise, gram negative cocci are typically round in shape, right? So gram negative cocci is typically N meningitidis, which is causing meningitis. N gonorrhea is also the second example, which is causing gonorrhea. The next case, if we want to look about, that is on the bacilli, that is gram positive bacilli, which is bacillus anthracis, called bacillus anthracis, which leads to skin infection and pneumonia. Likewise, Listeria monocytogenes may cause foodborne infections. Likewise, gram negative bacilli will also work. That is the best example, which is most commonly used and studied, E. coli. It can cause urinary tract infection. You have to take a note here that viruses do not stain with gram stain. Viruses are basically non-cellular components, but when they infect the host cell, they'll get activated and they'll serve like a living biological entity. They are not at all getting stained by these kinds of gram staining. There is a separate technique for the identification as well as testing of viruses. So this is all about the cell wall as well as the gram staining procedure. Kindly like the video, and if you like it, do subscribe the channel. Thank you so much.